A very good morning, everybody. I am Dr. Yukti Wadhawan from Delhi, and I'm going to talk about advanced paternal age. Does it really matter? It's been a long time since we've talked about aging. Now, is the aging a female only problem? This is the right time to talk about paternal aging and its impact on the offspring's health. So, is the male biological clock ticking? There was a publication in Human Reproduction in 2017 which studied the age of fathers in USA was rising. This study was done over a period of 40 years in more than 1.6 billion births, and they found that the mean age of fathers increased from 27.4 years to almost 31 years over the study period. They also found out that the percentage of fathers of newborns older than 40 and 50 years almost doubled. There was a Baltimore study on aging done on 890 men, published in 2001 by Endocrine Society, which studied that with increasing age of men, there is a longitudinal decline in the testosterone and free testosterone levels, resulting in high frequency of hypogonadal values. If you see this table, I have underlined in more than 46 years of age, there is a significant decline in the semen volume. If you see the bar diagram down there, the turquoise yellow and the sky blue, it also says that there was also a decline in the total motile sperm count with increasing paternal age. A similar study was published in a very important journal of andrology in May 2006. In the same year, Weirobek did a publication where he studied the relationship between male age and selected genomic defects in the sperm. If you see the first graph, it shows that with increasing age, there were more sperm DNA fragmentation and damage. In the second graph, you can see there were more achondroplasia mutations, FGFR2 and 113AGA, with increased paternal age. Interestingly, the increased age of the fathers did not affect the aneuploidies in the sperm. This was a publication in Fertility Sterility in 2008. And if you clearly see the pink, green, and red columns in the last two, so we see that there was, with advanced paternal age, there were more pregnancy losses and live births also decreased significantly. Now, last year, there was a publication in Human Reproduction Update, which studied the association of advanced paternal age and increased risk of spontaneous miscarriages. A month later, in June, another study was published, which said that there was an increased risk of spontaneous first trimester losses in men beyond 40 years of age. Now, if you see the statistical diagram in the last two categories, 40 to 44 years and more than 45 years, there was a significant increased risk of miscarriages in less than 20 weeks. In the 40 to 44 years, it was 23%. More than 45 years, it was 43% increased risk. Also, in the first trimester losses, in the same two categories, there was increased risk. And more than 45 years, it was a 74%, which is huge, risk of early miscarriages. Now coming to paternal age and aneuploidies. In 2017 in ASRM, there was a publication done on 243 egg donor ICSI cycles coupled with PGTA and NGS. This study found out there wasn't any effect of paternal age on embryo aneuploidy. Then in 2019, in Journal of Artificial Reproduction and Genetics, a similar study was published taking into account the blastocyst biopsy, and they too did not find any effect of advanced paternal age and embryo aneuploidies. Last year, a study was published in embryos derived from young egg donor cycles. So here, three groups were formed, group A, B, and C. A being less than 39 years, B was 40 to 49 years, and C being more than 50 years. They did not find any significant differences in terms of euploidy, aneuploidy, and mosaicism rates. Recently, in May, June, there was a meta-analysis published in Human Reproduction Update, which also studied the association between paternal age and aneuploidies in young donor oocyte-derived embryos. And they too found that there wasn't any association between paternal age and individual chromosome aneuploidies or segmental aberrations. Now, why are these studies important? They are particularly important for the fertility practitioners while counseling their IVF patients, and especially in couples where the older men, desire a child. Coming to the genetics. So, during cell division and replication, the genetic code needs to be copied exactly. This is all we know. So, if you see the picture on the left side of your screen, there is a mutated gene. The base pair is changed there. So, when such things occur, the coded instructions go haywire. There are faulty proteins, the nucleotides are in wrong order. As a result of all this, the body cannot function as it normally should. Way back in 1955, in the Lancet, there was a publication by Professor Lionel Penrose, and he found out that the paternal age and not the maternal age was predictive of de novo mutations. He proposed that mutations arose by DNA copy errors that accumulate over several replication cycles. Interestingly, spermatogonial cells replicate every 16 days, approximating 200 divisions by 20 years and 660 divisions by 40 years. Contrastingly, oocytes only undergo 24 cell divisions, that too till a woman's birth. Another study by British geneticist JBS Haldane said that children inherit more mutations from their fathers than their mothers. Now, what is paternal programming? So, due to the environmental factors, the lifestyle factors, toxins, smoking, obesity, all the hormonal milieu, abnormalities, all these insults affect the sperm during the developmental and maturation stage. As a result of all this, there is an altered gene expression in the zygote, which also causes impaired embryo growth and affects the health of the offspring. Now, if you see the graph there, down there, the maternal embryonic germline mutations, paternal, and also the offspring's post-zygotic mutations remain fairly constant throughout the aging of the parents. And paternal aging mutations per year were furthermore increasing than the maternal with increasing age of parents. Carrie Stephenson did a study in 2012 studying the de novo mutations, and he found out that the age of father and the mutation rate in offspring are strongly related. Dr. James Franklin Crowe has extensively done a lot of publications in terms of paternal aging, and he suggests that at present the greatest mutational health hazard is the fertile old males. Again, in 2017, Carrie Stephenson did an Icelandic study, and he said that there was increased number of paternal de novo mutations proband. Another study by Francioli was done on the same lines. He too found out that offsprings of older fathers harbored twice as many genic mutations as the younger ones. Advanced paternal age also increases the risk of autism and schizophrenia in the offsprings. Now, is man's biological clock ticking? If yes, then at what age? So this was a study of more than 25,000 men and recently published. It found out that in both the groups, donor and the patients, the shift to a significantly increased rate of DNA fragmentation index was found to be 41 years. And the overall changes in DFI and the high DNA stainability are driven primarily by age. Dr. Menezzo also did a study on clinical thresholds of DFI in 2017. So what he did was he formed three groups. One with a DFI less than 20%, they were the normal patients. The ones with DFI 20 to 25% were the gray zone, and more than 25% were the red zone. So he said, if you see the graph on the left, 20% DFI for early loss of fertility potential, especially if one or more classical semen parameters was compromised. 25% for natural and IUI cases, 40% for low probability for IVF ICSI success, and also in this group there was increased risk of miscarriages. So Dr. Menezes said that it is very important to supplement the patients in gray and the red zone. So anything with a DFI value more than 20, so they should be supplemented. So just briefly, taking you through this carbon cycle, if you clearly see in the middle, folic acid, vitamin B2, 6, 9, B12, and very importantly, zinc are required to complete one carbon cycle, as a result of which three major antioxidants, hypotaurine, glutathione, and coenzyme Q10 are derived. So why antioxidants are, and CoQ10 should be included in the protocol. So just getting you through this cascade, with increased paternal age, there is increased oxidative stress. Okay. So along with it, oxidative stress in the spermatozoal mitochondria induces lipid peroxidation, further increasing the ROS. Excessive amounts of ROS causes oxidative DNA damage, which is transferred to the offspring through fertilization and further increasing the risk of miscarriage, increasing the risk of autosomal X-linked dominant genetic diseases, neurological psychiatric conditions, childhood cancers, and more metabolic diseases. Coming to the conclusion, according to ASRM 2021 January, the expert opinion says clinicians should advise couples with advanced paternal age of more than 40 years that there is an increased risk of adverse health outcomes to their offspring. Data also indicates advanced paternal age increases the de novo, intra, and intergenic germline mutations, causing aneuploidy, structural chromosomal aberrations, and sperm DNA fragmentations. Genetic counseling may be considered for couples with advanced paternal age to discuss certain genetic risks in their offspring. The clinicians should discuss the importance of paternal structural autosomal defects in the evaluation of the couple with RPL and the need for the male partner to have a karyotype analysis. Given the increased risk of miscarriage for men with abnormal sperm DFI, testing for the DNA fragmentation is also indicated in males wherein the couple is suffering from RPL. Thank you.